So my name is Maureen Kachman. I am the managing director of the Metabolomics course. So along with Dr. Brandt, who's our faculty director, I sort of manage the lab, talk to people who are trying to bring samples to us, do my best to offer advice as to how to do your study design, but often bring in a lot of other members of the core, Dr. Brandt, Charles Evans, any of the people who are actually running your samples because they have a lot of great information, Lee Zhang, who might or may not be here. And especially when it comes to different matrices and things like that, you'll often hear me say, well, let me check with somebody who's done that before, because the cores run thousands and thousands of samples, but you may have something that we haven't done before. I mean, we're always learning and we're always doing new things. So today I'm talking about just interacting with the core, like how do you get started with us? And again, just kind of reiterating some things about how we sort of divide the world into targeted versus untargeted analysis, and what happens if one of our targeted analyses doesn't work for you, you're looking for something specific, how do we do method development and things like that. So briefly, how do you procure services from the core? I'm not going to go into a slide-by-slide, presentation of like how you go and click on, an assay in our system. Our sample manager's name is Carrie Bonds. Some of you have worked with her. She is really good at getting you through that system, although it's a little clunky still. And she also, I think, will talk with you, especially if you're from out of the University of Michigan, about getting a head start on some of the paperwork and things you have to do, because there is a little bit of a process to get started with us for the first time. So if you're thinking about doing samples with us and you're outside of the U of M, especially, contact us a little bit in advance, because there are some agreements that have to be done between institutions and things like that. But she's good about getting us through that system as quickly as possible. But please do talk to us at the earliest possible time if you're considering sending samples to us. We're also going to talk about standard offerings, like what we have, assays we've already developed and why we do them and what they involve. And then also, so to understand the role of data interpretation for untargeted metabolomics, I actually have a whole talk about that, as do several other people have bits and parts on Wednesday. So I actually won't talk about that too much today. Okay, so Kelly didn't speak today. Anna took her part of the presentation, but Anna talked a lot about some of these pre-analysis considerations that you need to think about. Sample collection, she talked a lot about the biological background of sample collection, making sure that you're careful with how your animals are fed and the time that you're collecting the data and such. I also want to bring in from a mass spectrometrist point of view the idea that if you ever think that you're going to have some kind of unusual sample that's coming to us, talk to us a little bit ahead of time. I'll give you an example. I had a customer who came and they wanted to do skin swabs, and this is great, except for they were swabbing the skin with some, two different kinds of swabs. One of them was cotton-based, like a Q-tip, and the other one was like foam. And kind of a nightmare for metabolomics, because anything with foam in it also has a bunch of other small molecules in it, polymers to be exact. So those are not the optimal collection type of vehicles for metabolomics. So anytime you think you're going to want to start a study, please talk to us about things like that. Yes. Okay. Okay. Yeah, I mean, so if it was me, I apologize. There's a bunch of us who are taking email requests from people on a daily basis, and I try very hard to get to my email queue every day. So if I missed it, I'm sorry. But yeah, just talk to me here at the workshop. That would be actually great. And then phone call is always good too. Yeah, thank you. Yes. There's more. I'll go through a little bit more about the website. So, okay. So she talked about washing and quenching of samples. Oops, I'm sorry. Pointers up. Again, I'm going to point, and I believe, did Alice say that we put a copy of our sample collection guidelines in the folder for everybody? We did. Okay, so just that is about a 15-page document that talks about how to collect tissue, how to collect fecal samples, how to collect saliva, how to collect cells. The washing and quenching part is obviously important. Charles talked about, why you want to quench your metabolomics very quickly and make sure that you stop, enzymes from continuing to act on your samples. Washing is also important, again, in that guidelines system, in that guidelines document, because you want to make sure that you remove as much of the media as possible. Otherwise, you're going to be getting metabolite signals from anything that's in your media, that is not endogenous to your cells. So she talked a little bit about the use of biological replicates. I won't go into detail there. And the collection and control of metadata. Again, I won't go into detail with that. Okay, this isn't really working. I'm just going to skip this slide. You guys have seen it before. But it's definitely an iterative process to talk about metabolomics and to do consultation and to receive your data and then to evaluate your data with us. So Charles talked about targeted versus untargeted metabolomics, and how there's a blending of those two worlds. But you might think of targeted metabolomics as being hypothesis-driven metabolomics. In other words, you really kind of know or have a good idea which pathway you're interested in, and you want to measure particular metabolites within that. This is an example of some work that was done at the core. This particular investigator had a hypothesis that that kynurenine was impacting the presentation of or the severity of C. difficile infection in his mice. So after doing a number of other studies, he decided that he was ready to do the knockdown and knocked out the IDO1 gene. So, and then brought the samples to us and had us measure the tryptophan pathway, and found that his knockout did in fact work, and that in the wild type mice, there was a normal amount of kynurenine, but in the knockout here there weren't, and in fact was able to therefore go back later and associate with that abnormally low amount of kynurenine with the severity of the C. difficile infection in the mice, and then with many more steps later come up with the mechanism for that. But this was a method development he asked us to do, and we were able to do for him. So, again, this is what our website looks like. You'll see up at the top services and fees. And this is what I'm going to show you on the next slide, which is that we do list all of our standard assays here. They're broken out by targeted, untargeted, and lipidomics, for example. So you kind of have to look in a couple of different places to find what you're looking for. But we did that on purpose because a lot of times people who are interested in lipidomics really want to see all the lipidomics offerings in one place. And if you're interested in targeted metabolomics, you probably just want to see what the different pathways that we measure are. We do have a sort of a PDF here describing each of the assays that we have come up with here in the core. I just want to bring your attention to what that looks like. If you download that PDF, there's some really important information on there. The metabolites that will be reported, the limit of detection and the limit of quantitation, and then the amount of material that we need to do the assay. Really important that you check that because it's different for every assay, and it's very difficult to do like in a cosanoids platform if you only give us 10 to the 5th cells when we're asking for 10 to the 7th. So because those are lower level metabolites, so we're going to need more cells to see that. What was I going to say about that? Oh, and then again, the limit of detection it's again different it's the same for the assay. It's it's based on the instrumentation that's used to run the assay, but it's different for for you, from your point of view, for each type of matrix that you're using. So you want to kind of have an idea ahead of time, what are the expected levels of these metabolites in cells or in tissue or in plasma. To some extent we can advise on that because we know, well, we have this eicosanoids platform, but it's, these eicosanoids are much more abundant in tissue, and these ones are, maybe, less abundant in plasma and a little bit more difficult to see. So we can kind of let you know ahead of time, whether you're going to see everything that's available in this platform or not. But again, there's resources that I'll talk about later for you to kind of go back and do some research to see, if this is the best way for you to approach this problem, and whether you're likely to get a positive result or not. The other point I was going to make about that was that we are constantly, obviously, working on the assays, trying to improve them. And the most improvement we've had so far is in the volumes, because we have a lot of people dealing with rodents. So a lot of the volumes that are on here probably need to be updated. We can do a lot of stuff with rodents in about 20 microliters of plasma, for example. But don't just assume that, because we may need more for certain assays than other ones. Again, we're willing to try to do it with a lower volume than what we've asked for. It just means that we probably have less options if something goes wrong. In other words, if we are going to extract 20 microliters of plasma, we only may get one shot at it in the mass spectrometer, as opposed to having the ability to do multiple injections of that metabolite, or of that sample. So targeted assays generally, we're going to try to map as much of a particular pathway or a compound class as possible. Sometimes we need to use multiple methods. So, for example, for glycolysis and TCA, in the TCA cycle, it's fairly easy to see citrate, succinate, and malate by LC, but alpha-ketoglutarate really doesn't show up really well in the LC. It's much better done by GC. So that can get very time-consuming and can be expensive. So it's also probably a good idea to think about, what are the critical parts of the pathway? What do you really need to see? Again, we're willing to do both, but if just doing one is better, then we'll just do one. Generally, we provide absolute quantitation. Now, I'm going to get into what that means exactly in my next talk a little bit more. But again, targeted assays will generally give you some value that is in, if it's plasma, it would be micromolar, for example, or nanomolar, or whatever it is. And if it's in tissue or cells, we're going to normalize it to the tissue amount or the protein content of the cells, so it would be like micromoles per milligrams or whatever the case may be, as opposed to untargeted metabolomics, which is really just kind of looking at peak areas and doing a relative quantitation. And again, this was actually touched on earlier today a little bit about normalizing as well, and I'll talk about that in more detail next time. Okay, so you come to our website, you see some standard assays that we offer, but you've been reading the literature and you've been saying, well, there's some other metabolites that I think are going to be important here. We are always doing method development. We have a couple criteria we use for method development. One is, if we think a number of people are going to be using it, then it's definitely worth our time to invest in doing that. And there may or may not be costs involved. Usually there are costs involved, but if it's a very, very specific method development that you need to have done, then there'll probably be some costs for both the recovery of the standards that need to be bought and the time. Again, these are things where if it's something that we think a lot of people are going to use, we'll work with you on that. So, but what are the steps for doing method development? So just so you guys know. The first is just, we would do a literature search. We would say, okay, has anybody done this before? That involves, kind of looking at the methods critically and saying, well, that seem to work, but it's got 75 steps and probably isn't as reproducible as you'd like it to be in a core facility setting. Or do we have the same instrumentation that was used in that method that was published? Often we can modify it and do something similar with what we have. If there's nothing and we don't have a clue, then we will go ahead and try to start with our own method development from there. And one thing I want to point out about that is that Steffi, who talked earlier, has done a great kind of thing for the core. She actually did a comparison of about 8 different stationary phases, including reverse phase, and like 4 kinds of Hillock and such. So we actually have 850 or maybe 1,000 now standards in our metabolomics library. These are, powdered standards from Sigma Aldrich or whatever. And she ran those all against all these methods. So if you're looking for something particular, we probably have a good idea already of what kind of chromatography we want to use to look for that. So we use that as one of our starting places in terms of deciding how we're going to, first of all, separate, the metabolites of interest. And we'll go on and talk about, but assuming that, again, if we don't have that in our library, if it's not one of our 1,000 compounds, the next thing we'd have to do is purchase an authentic standard. You'll see a lot of stuff in the literature where people are not necessarily doing that. They're just sort of guessing as to what the identity of the metabolites are based on either accurate mass or, MSMS, which Charles talked about, is a powerful method for identifying compounds, but it's not really a substitute for having an authentic standard. There are times when you can't get an authentic standard, that nobody makes it. You can ask for a custom synthesis from a number of different manufacturers. Tends to be very expensive, $3,000 to $10,000 in that range. So if we can't get an authentic standard, then we have to decide what to do next. But we would always try to get an authentic standard if possible. And then once we decided that we think we know kind of maybe which type of separation method to use, and we're able to procure the standards of interest, the next thing is just to decide what kind of mass spectrometer to use. Again, this is where the challenge comes in, because if you have, you're looking for a broad sort of class of compounds, we might need to use multiple methods. But we would develop a method that was either tough, so based on just the accurate mass, or if we need a little bit more sensitivity, we'll use triple quad. So that will get us more sensitivity, but I'll talk about what we have to do to get a triple quad method in place a little later too. Or GC if it's something that is a compound that can be volatilized and is best detected that way. So this looks great, like the first 4 or 3 steps on standards. But after that, you still have to think about how are you actually going to get the metabolite out of the tissue or the cells of interest. And so the next thing that comes into play is to really look at what the homogenization and extraction method will be. So, in most cases, homogenization isn't terribly difficult. We have different methods for cells, which just mainly involve like manually scraping the cells, and tissue, which involves either using, we have a couple different ways we do tissue. One would be to use sort of a probe sonicator. So that would be a probe that comes down into the Eppendorf tube with the sample in it and sort of blasts the sample apart. For really tough tissues, you would actually probably want to freeze that in liquid nitrogen and grind it into a powder, and then try to quantitatively transfer that to another vial so it can get extracted at that point. So those are all important considerations, usually not too difficult for most human or cell lines, challenging for fly heads, which I just talked to one of our friends about today. The extraction method, again, we have an idea for the most part of, the types of solvents that we need that will be able to, extract the compounds of interest based on their polarity for the most part. But finding the optimal extraction method can be a little challenging if you're looking for, both polar and nonpolar compounds in the same analysis. Metabolite stability. How well are those metabolites going to survive in the extraction solvent of interest? An example of where this really comes into play is like thiol compounds are pretty unstable. They can, polymerize very quickly or get converted into something else. So what you want in that case, if you're doing thiols, you almost have to like run them the same day as you extract them. So there's just considerations such as that we need to worry about. And then again, what I talked about a little bit earlier, like if we want to make sure that the method that we're going to be developing, once we got it down low enough on standards to the, whatever it is, nanomolar level or whatever, that, we have a good chance of seeing that in your compounds anyway, that we've done some literature searching to see sort of what's the expected concentration in your sample. I mentioned HMDB. I don't know probably if anybody here is familiar with that. Can't remember if I talked about it in one of my later talks, but it's called the Human Metabolome Database, and it's a great resource for sort of going in and looking at metabolites and saying where has it been seen before, what kind of tissues, urine, plasma, what are the levels. It's really interesting because they actually have in there normal and abnormal levels as well. It can give you some ideas, like if people have studied normal human beings in certain age ranges, what their levels are, but other studies that have reported it, and for example, people with a certain disease state, they might have very different levels, so it's important to make sure that you're paying attention to whether you're looking at normal or abnormal levels if you use that resource. All right, so just to show you a little bit about method development, this is a pathway that we've developed an assay for. It's a little bit challenging because most of the compounds here have some pretty different qualities. Some of them are pretty hydrophilic with the diphosphates. Others are a little bit more hydrophobic with the CoA. And so creating a method for this was a little bit challenging. So the first thing we did was we bought all the internal standards, or the authentic standards, I should say, and just kind of decided whether we could separate them chromatographically, which we were able to do. And keeping in mind that there are compounds that can be isobaric, have the same mass, and just have a different arrangement of their atoms. So this is an example too, where two of the compounds in this pathway are isobaric, but they separate very nicely on the HPLC. And again, this was done by sort of a TOF instrument, so we're able to just sort of see the mass of this ion, and I'll explain a little bit more about why we might want to look a little further at these. There are some other things that come into consideration. Some ions, even though we can see them at a particular mass, they may like to do things such as kind of decide to show up at half their mass because they pick up 2 charges in the mass spectrometer. So those are called 2+ ions. So if the 2+ ion, so this is the parent ion here, and we get one that's about half its mass, if that one's more sensitive, then that's the one we're going to monitor for. So we always keep an eye out for things like that and try to figure out, what's the most sensitive peak that we can monitor for. And I think that was it for this particular slide. Oh, okay. So I did talk about HMDB. Sorry. Again, this is just, if you haven't used HMDB before, it's just a great place to go in and see where this metabolite's been detected before. and then again, go back and get some sort of normal or abnormal levels just to see if you can say that it's likely that this compound is going to be in the range that we're able to detect it based on what we can see in our standards, for example. So, so we thought when we first did this analysis that we were going to run this on triple quad based on the information that we saw from HMDB, that although the TOF looked pretty good on the standards, they just weren't, it just wasn't getting low enough for what we needed based on what we could see in HMDB. So we decided to go ahead and run this on a triple quad, and to our surprise, the first time we did this, the sensitivity looked a lot worse. And so this is something that happens occasionally because you start thinking about a few things, like how is the front end of this instrument different from the front end of the TOF, and how, you know, how do we need to look at the energy of the molecules that are getting into the mass spectrometer. And this, I think Charles alluded to this, that they were getting a little too much energy and we're sort of trying to break down before they even got in the mass spectrometer. So that was just something that was, it took a little bit of time to work out what the conditions were for the front end to get this to really be truly, you know, more sensitive than the TOF instrument that we had originally used, and to make sure that we could detect it. Another feature I want to point out about when you're using a triple quad, Charles described what a triple quad does, but essentially what you're trying to do here, which can be a really powerful thing for doing targeted metabolomics, is to look at a particular parent and fragment ion combination, and that's called an MRM for those of you who have heard that term before. And the reason this can be very helpful is because when you have isobaric compounds, if you don't get a perfect separation in the LC, you might be able to find one fragment that is unique to one of the compounds that, you know, you don't see it in the other. They're the same mass, but they fragment a little differently. So you can find this one fragment that you can monitor for that uniquely identifies one of the two compounds. And when you do that, again, so you want to balance that. You need to have that unique signature of that compound, but you also want to quantitate that compound well. So if it produces a unique signal, but it's very low intensity, then you have to make a compromise. And the compromise is usually pretty straightforward. You just say, I'm going to use the unique peak to identify the compound, but I'm going to use a different peak, which is more intense, to actually quantitate the compound. And so you might have two MRMs for the same compound in that case. So if you have questions, you guys can stop me. But yeah, this is Hillick. Yeah, yeah, and I can tell you that details later. So okay. Sometimes when you're doing that method development and you're trying to find those unique fragments that that uniquely identify a particular compound, compound, you can do an experimental version of it where you let the instrument sort of kind of go through and fragment and then start to pick the fragment of interest and sort of start to optimize around it, and that works great. But sometimes it doesn't work as well as you'd like it to, and so I just wanted to, you know, bring in that sometimes we'll use another resource here where we'll actually go before we start doing the development to figure out what that unique fragment is. We will take a theoretical database and put in the structure, and then say, can you predict based on the chemical bonds what the most intense fragment will be, or what all the fragments will be, and see if we can find the one that is going to uniquely identify that compound. And I've actually found this to be very helpful, and especially because it sort of like predicts, you know, what your relative intensity is going to be. So you can say, I definitely don't want to use that one. It's way too low. So this is just a nice resource for people. This is, I believe, the name of this program is called CFMD, but it was written by a group out of Canada called the Wishart Lab, which actually also runs the HMDB. Okay, so once we've decided that we either have the MRM or the TOF method down, we're going to want to decide on a series of calibration standards that we think are going to bracket the concentrations that will be in your samples, and we will do that. We'll basically do 5, 6, 7, 8-point calibration curve, whatever we need to do with authentic standards. Oh, okay, sorry. One of the things that's important about a calibration curve is that, I'm sorry, we do generally tend to do a calibration curve for every metabolite in a targeted analysis, but you would also like to have internal standards for each of the metabolites if possible, because you want to be able to just make sure that from one run to the next that the instrument's behaving reproducibly. And so the internal standard will kind of help with that. I guess I'm actually going to talk about that in my next talk. So I'll go into more detail on that, but having an internal standard also goes into how we quantitate the samples that before they get onto your calibration curve. Actually, since I am going to talk about targeted metabolomics in a couple days, and you'll hear more about it over the next couple days from several other speakers, I think I'll just stop here with this particular talk. Okay, so again, interacting with the core, since that was the purpose of this talk, I do want to say that it's important to consult with us. People, again, have gotten a little basic understanding today of like the difference between untargeted metabolomics and targeted metabolomics, and then this third category, which we call fluxomics or stable isotope labeled metabolomics, and we kind of think of that as a separate entity. But I just wanted to point out that the way we think of fluxomics is that really any targeted assay that we have at the core can be turned into a flux assay. So in other words, if you can figure out how to label your sample with a stable isotope, then we can probably take any targeted assay we have and just modify it to monitor for all of the isotopes that will be produced by your label, as opposed to just the main isotope or the main compound of interest. So as you're thinking about as you're listening to flux talks over the next couple of days, and you're thinking about different pathways for flux, and if you ever go back to our website and you say, oh, those would be really interesting things to measure. Just keep in mind that pretty much any targeted assay we have can be turned into a flux assay, more or less. So, all right. Well, I guess we're going to go on to my next talk today, which is supposed to be tomorrow, but you guys will get a break shortly. So hang in there with me for a few minutes, okay? Yeah. Any questions about that? Yes, there we go. Okay, thanks. Okay.